Welcome to the deep dive. Today, we're really getting into something fascinating, CRISPR gene editing.
hmm
It's this technology, that lets us make these super precise changes to DNA. It's caused a huge stir.
Oh, absolutely. A massive stir in biomedicine. Yeah.
And you, our audience, sent over some great sources. So our plan today is basically to pull out the most important, most interesting bits, make it clear, easy to follow.
Sounds good.
We've got an interview with Stanley Chi, a bioengineer from Stanford, and also a really good piece from Technology Networks covering the latest advancements.
Both excellent sources. Lots to unpack there.
Totally. So let's just dive right in. What exactly is CRISPR? Where did it come from?
Well, it's actually kind of a wild story. Its origins are surprisingly in microbes.
Microbes, like bacteria.
Exactly. Bacteria and archaea. The basic sequences, the CRISPR parts, were spotted way back in 1987.
Wow, okay.
Nobody really knew what they did until maybe around 2005. Turns out it's their immune system.
An immune system for bacteria.
Yeah. A defense mechanism. It's pretty amazing that this powerful tool we're using now started out like that. Nature's efficiency.
That's incredible, like a tiny security system.
Yeah.
And the name itself, CRISPR. It sounds complicated.
It is a bit of a mouthful. Clustered, regularly interspaced, short palindromic repeats.
Right.
But it basically describes this pattern in their DNA. Think of it maybe like a genetic scrapbook.
A scrapbook. How so?
Well, if a virus attacks the bacterium and the bacterium survives, it can actually grab a little piece of the virus's DNA.
Seriously?
Yeah, and it stores it right there in its own DNA in those CRISPR sections.
Okay, got it. Storing evidence.
Precisely. And then it uses that stored viral DNA to make something called guide RNA.
Guide RNA.
Uh-huh. Stanley Chi made a good comparison to our own immune system, like how our antibodies remember germs.
Yeah,
This guide RNA is like that genetic memory. So next time the same virus attacks.
it recognizes it.
Exactly. The guide RNA spots the matching viral DNA sequence, grabs onto it, and then guides a CRISPR protein, often it's Cas9, right to that spot, and
So-
cuts it. Destroys the invader's DNA. Super targeted.
Wow. Okay, so it's like this incredibly precise molecular guided missile system,
That's a great way to put it, yeah.
which brings us to the big question, right? Why is this such a massive deal for us? I mean, haven't we had ways to edit genes before?
We have, but that's the key difference. CRISPR's precision is just unprecedented,
Yeah.
and it's relatively easy to direct it.
Easier than before.
Oh, much easier. We can point it to specific DNA sequences in our own bodies, or even tweak the chemistry around the epigenetics, as Dr. Key called it.
Hmm.
And this just opens up amazing possibilities for treating, maybe even curing genetic diseases. Things where, regular drugs or surgery just can't fix the root cause.
Getting right down to the genetic code itself.
Exactly.
Well, we're actually seeing this happen now, right? That Technology Networks piece mentioned the FDA approval for Casgevy for sickle cell.
Yes. Casgevy. That's a huge milestone. Sickle cell anemia is, well, it's a perfect example.
Why is that?
Because it's caused by just one tiny mistake. A single letter typo in the gene for hemoglobin.
Just water.
One letter difference, and it caused all these terrible health problems. Red blood cells change shape, block blood flow.
Right.
What CRISPR allows, in this case with Casgevy, is to go in and precisely correct that single faulty bit of code.
Instead of just managing symptoms.
Exactly. Current treatments, like Dr. Key mentioned, are often transfusions, bone marrow transplants, tough procedures. Casgevy offers this hope of a one-time fix.
A permanent fix.
Potentially, yes, fixing the underlying genetic problem. And think about it, there are over 8,000 known genetic diseases.
8,000. Wow.
Yeah. So the potential impact is just staggering. It's kind of mind-blowing that fixing one letter can change a life so drastically.
Like, it really is. Puts the power of this into perspective. Okay, so you said CRISPR is way more precise and easier than older methods. How did those older ones work?
Well, the older tools, things like zinc fingers or talons, they were basically protein-based.
Okay.
You had to design a whole new protein, engineer it very carefully every single time you wanted to target a different gene sequence. It was incredibly slow, very laborious, and expensive.
It's like building a custom tool from scratch every time.
Pretty much, yeah. CRISPR is different because its targeting relies mostly on that guide RNA molecule.
The bit that matches the target DNA.
Exactly. And making a specific RNA guide in the lab. That's relatively quick and cheap.
Ah, I see.
So Stanley Chi's analogy was like a GPS, right? With
Yeah.
the old tools, it was like rebuilding the whole GPS unit for every new destination. With CRISPR, you just type in a new address, the new RNA guide, and the system, Cas9, usually knows where to go.
That makes so much sense. Much more programmable.
Hugely. It's faster, cheaper, more precise, just a massive leap forward.
Okay, so it's powerful, it's precise, but the Technology Network's article talks about challenges too, like actually getting CRISPR into the body, right? Delivery.
Yes, delivery is definitely a major hurdle. The article mentions two main strategies, ex vivo and in vivo.
Ex vivo and in vivo.
Ex vivo means outside the living body, so you take cells out of the patient, say, blood stem cells.
Like with Casgevy.
Exactly like Casgevy. You edit the cells in the lab, make sure it worked, and then you infuse them back into the patient.
Okay. Sounds complex.
It is. It works, but it often means hospital stays, complex procedures, and yeah, it's expensive. Casgevy, as they noted, is about $2.2 million per treatment.
Whoa. Okay.
So the other approach, in vivo inside the living body, is the goal for many therapies. That means delivering the CRISPR machinery directly into the patient's body to edit cells right where they are.
That sounds much more direct, less disruptive.
Potentially, yes. And likely cheaper and more accessible if we can get it right. But it's tricky. You need to get it to the right cells and avoid hitting the wrong ones.
And that's where Jennifer Doudna's lab comes in, with these EDVs,
Right.
enveloped delivery vehicles.
Oh, EDVs. It's a really clever idea. They're basically using parts of viruses, just the harmless outer shell, like an empty delivery van. They load the CRISPR components inside, then they decorate the outside of this van with specific antibodies.
Antibodies, like from the immune system.
Exactly. Antibodies that are designed to recognize and stick only to certain types of cells, like T cells, for instance.
So it's like putting a specific address label on the delivery package.
That's a perfect way to think about it. It targets the delivery just to the cells you want to edit.
And they showed it worked in mice, targeting T cells, but not liver cells.
They did, which is really promising for specificity. And they even used it to create CAR T cells directly in vivo.
Okay, SAR T cells. I know that's a big thing in cancer therapy, but
Yeah.
can you quickly remind us what it is?
Sure. CAR T-cell therapy takes a patient's own T cells, which are immune cells, and engineers them in the lab to have a special receptor, a chimeric antigen receptor, or CAR.
Right.
This CAR helps the T cells find and kill cancer cells much more effectively.
But usually that engineering happens outside the body, right? The ex vivo way.
Yeah, correct. It's a complex, expensive ex vivo process currently. Being able to create those CAR T cells inside the body using something like EDVs, that could be a huge game changer. Make it simpler, cheaper, available to more people.
That's amazing. So CRISPR isn't just fixing genes, it's also being used to, well, study things in new ways, like this CHIME system for the immune system.
Yeah, CHIME chimeric immune editing. That's another really smart approach coming out of Harvard, mentioned in the Technology Networks piece.
What's different about it?
Instead of editing mature immune cells, CHIMO goes earlier. It targets the stem cells in the bone marrow before they become different types of immune cells.
The precursors.
Exactly. They take out these hematopoietic stem cells, use CRISPR to edit specific genes they want to study, and then put the modified stem cells back into mice.
So they can see how that specific gene edit affects the whole immune system as it develops.
Precisely. It lets you study gene function and immunity without some of the artificial effects you might get from editing mature cells in a dish. You see the long-term impact.
And they've made it even more sophisticated. XCHIM.
Right. They've developed variations like C-CHIME, I-CHIME, L-CHIME, S-CHIME, collectively X-CHIME, allowing more complex studies.
Like editing multiple genes at once, multiplexing.
Yep, multiplex editing. Or even making the gene disruption inducible, meaning they can trigger the edit to happen only when they give a certain drug. Gives them more control.
And they use barcodes, genetic barcodes.
Yeah, that's super clever. They add unique DNA sequences like tiny molecular tags to the edited stem cells.
So they can track them.
Exactly. They can follow the descendants of each edited cell, see how much they multiply, where they go in the body. It gives this incredible resolution for understanding immune dynamics.
Wow. The applications just keep branching out. The article also mentions synthetic biology, CRISPR copies for making biofuels and stuff.
Right. So that's moving beyond medicine. CRISPR copies is a computational tool. It helps scientists figure out the best spots in the genome of microbes, like yeast, to insert new genes for metabolic engineering, especially useful for yeasts that aren't the standard lab models.
So making them produce useful things more efficiently.
Exactly, like finding the optimal place to add genes so the yeast overproduces something valuable, like 5-aminolevulinic acid, which they mentioned is used in agriculture and food. It speeds up that engineering process.
Makes sense. And plants too.
Yeah.
Improving crops.
Yes, another big area. Researchers are working hard to get CRISPR working really efficiently in plants for multiplex editing, changing multiple genes at once.
To get better traits faster.
That's the goal.
Yeah.
The work mentioned at Bioenginee in Belgium, they're tweaking the CRISPR system components, things called promoters, which control gene activity, and NLSs, which guide the machinery to the cell nucleus.
Little tweaks to the system itself.
To get much higher success rates for making multiple edits simultaneously in plants like Arabidopsis, this could really accelerate developing crops with, say, better yield or disease resistance.
Okay, one more application from the article, mapping cancer, using CRISPR barcodes again.
Yeah, the EvoCat model. Similar idea to the immune cell tracking, but applied to cancer.
How did that work?
They used CRISPR to put unique barcodes into prostate cancer cells in mice. Then they could track where those cells spread metastasis.
To the liver, lungs.
Right. And they found some interesting things, like often the spread originates from just a few really aggressive clones.
Not just any cell.
Seems like a small subset drives it. And also, once cancer cells set up shop in a new organ, they tend to stay there rather than spreading again immediately from that new site.
That detail must be really useful for understanding how metastasis actually works.
Absolutely. It could help design better ways to stop or treat cancer spread.
Okay, so we've talked about fixing diseases, studying biology, engineering microbes and plants, tracking cancer. It's incredibly powerful, which inevitably brings up the ethical side, doesn't it?
It absolutely does. You can't talk about CRISPR without touching on the ethics. It's crucial.
Stanley Shi mentioned it briefly, too.
Yeah.
Things like designer babies, editing human embryos.
Right. That's probably the biggest red line for most people. Germline editing, making changes that would be passed down to future generations, is widely considered unethical right now. The potential risks, the unforeseen consequences.
Yeah.
And then there's the spectrum of use, right? Using CRISPR to cure a devastating genetic disease like sickle cell, most people see that as a clear good.
Makes sense.
Using it for prevention, say, editing a gene that slightly increases risk for a disease later in life. That gets into a gray area.
Yeah. Where do you draw the line?
And then there's enhancement. Using CRISPR not to fix a problem, but to try and make someone better, smarter, stronger, taller. That raises huge societal questions.
Like who gets access? Would it create even bigger divides between people?
Exactly. Equity is a massive concern. And there are ecological worries too, like using gene drives based on CRISPR to wipe out species like mosquitoes. Could it have unintended impacts on ecosystems?
Lots of big questions we need to grapple with as the technology advances.
Definitely. The science is moving fast, but the ethical and societal discussions need to keep pace.
So looking ahead then, putting the ethics aside for a moment, what does the future look like? What did Stanley Key envision?
Well, he talked about improving quality of life during aging. Maybe not making us live dramatically longer, but healthier for longer, mitigating some of those age-related declines.
That sounds appealing.
And he mentioned engineering organisms for sustainability, microbes capturing solar energy maybe, or developing much more nutritious food.
Using CRISPR for energy and food security?
Yeah. And then the really ambitious idea, a universal vaccine.
Against viruses.
Potentially, yeah. Using CRISPR's original function, that bacterial immune system, to somehow target and destroy a wide range of viruses. That's definitely more long-term, a big research goal.
Some things feel closer, like the therapies, while others are still way out there.
Exactly. The therapies for monogenic diseases, the ones caused by single gene errors, are happening now. The other stuff.
Right.
It's exciting, but still requires a lot more work.
It really is stunning, though, how far CRISPR has come so quickly. I mean, that key paper was only 2012, right?
That's right. Doudna and Charpentier's paper. From that discovery to FDA-approved drugs and a Nobel Prize in about a decade. It's just remarkable speed for science.
Truly is.
And as both sources really highlight, even with these huge milestones, it feels like we're still just scratching the surface. The research is exploding in so many directions: basic science, medicine, agriculture, diagnostics.
Yeah.
It's a very dynamic field still.
So it's clear this technology gives us this amazing, almost scary level of control over, well, the code of life itself.
Mm-hmm.
Thinking about how fast it's moved already. What kinds of things that seem like science fiction now might actually be possible in, say, the next 10 years? And maybe more importantly, what kind of responsibilities come with having that kind of power?
That is the question, isn't it? A lot to think about.
Definitely. Well, thanks for taking this deep dive with us today. It's been fascinating.